Hello and welcome to this video. I'm Ian Hoffecker, and this is another lecture on machine learning in biotechnology. And in this second series of lectures on this topic, I'm going to be talking about probability theory. And the reason we're talking about that is because in the last lecture, we talked about how machine learning is actually a form of function approximation. And approximation is going to be inherently probabilistic. And so to kind of recap what we said, we have some sort of function that's mapping an input x onto an output y, or transforming in some way an input to an output. And while you could have a mathematical or computer function performing this task, in many cases, this is actually going to be a natural physical process that's doing this. And the goal of machine learning in many cases is going to be coming up with a function that can take an input and figure out the correct y output without explicit knowledge of the underlying function or physical process governing that transformation. But the key word here is approximation. And so since we're dealing with uncertainty, this leads us to the realm of probability theory. And so in many cases, we'll be working with statistical models to represent data. We'll be working with neural networks that are trained with probabilistic loss functions, or we'll be looking at the structure of data using unsupervised techniques using probabilistic statistical models. And so some topics that will come up over and over again are the idea of uncertainty. So how confident are we that a model has given a particular classification result? Generalization. So our goal is to have some input data, but be able to move beyond that data and deal with future data, which is inherently uncertain. We'll be using loss functions or optimization functions, many of which are inherently probabilistic, such as the likelihood function. And then lastly, motivation here for probability theory is if we open up a machine learning paper. So this is a screenshot from the AlphaFold paper. So this is, of course, big news in biotechnology, the ability to predict the protein structures of many different proteins. And if we want to try to understand what exactly these authors did here, we'll immediately start encountering words like confidence, Kullback-Leibler, divergence, distribution, and we might see something like this. So this is a conditional probability statement here. And so in order to read machine learning literature, we're going to have to be literate in the language of that literature, which is probability theory. So we'll start from the beginning. Where does probability come from? What is probability? And so if we go to a familiar example, let's say we're flipping a coin, and it's a perfectly fair coin, let's say 50-50 chance of heads or tails, and we flip it 4 times. So what kind of result are we expecting? Or how many heads do we expect to get? So let's say we flip it once, we get heads. Flip it a second time, we get a tails. Third and fourth time, we get another 2 heads. So we got 3 heads in the end. And kind of silly question maybe I can ask is this a surprising result? And so there's a couple ways to answer that question. But another sort of naive question we might ask is, why don't we always get 2 heads and 2 tails, right? This is supposed to be a perfectly fair coin, 50-50. So why did we not get a 50-50 result in the end? Where does this come from? And so to kind of build some intuition there, it's instructive to map out all the possible events. So if we flip a coin once, we have 2 possibilities, 1 heads or 0 heads. If we do it 2 times, actually, we see that now there are 4 possibilities, because we can get 2 heads, or we can get 1 heads and 1 tails, or 1 tails and 1 heads, or 2 tails. And we can do this for 3 flips or 4 flips. And we'll start to see that the space of possible events, so we'll call this the sample space, begins to take on this bell-shaped structure. And one of these results here we might call an event. And so if we start to formulate this in the language of probability theory, we might assign a variable to that event. So we might say the variable A here represents a particular event, say the HHHH event. And then we can ask the question, what is the probability of A, or what is the probability of this 4 heads event? And the easiest way to do this here, or an intuitive way to do this here, is to simply count the ratio of the event that we've determined to the number of total events in the sample space. So there are 16 possible events, and there's one HHHH event. So we can compute the probability from this fraction here. And we can do the same thing for the case where if A here represents H is 2, or 2 heads. So then that would be this middle column here. And we would see that there are 6 possibilities in which there are 2 heads emerging. And once again, still 16 possible events. So 6/16 then becomes the probability for getting 2 heads. So if we go back to this question, is this a surprising result? So in some sense, it's not so surprising. We can see that there are actually multiple ways to get 3 heads. So there are 4 ways to get 3 heads. And 4 out of 16 is not a totally unreasonable result. So this way of thinking about probability in terms of events, turns out this is the foundation of probability theory itself. So all of probability theory is built upon these 3 basic axioms, the axioms of probability. And so we'll use this term, again here we have a variable A. So this is some binary variable that is either true or false. So for example, are there 2 heads? And 2 heads is going to be true for these 6 cases here, and then false for the other cases. So that could be our variable A here, and then the sample space would represent all the other events. And the probability axioms then are going to be the following. So the probability of our event A must be greater than 0. The probability of the sample space, so all possible events must sum to 1, must be 1. And the probability of any 2 events, any of either of 2 events, so A or B, or A union B, where A is one event and B is another event, this is going to be equal to the sum of those 2 events, A and B probabilities, minus the probability of the intersection of those events. So intuitively, you can picture this as, if this represents A, and this represents the space of B events, and if they have some overlap here, we're going to count A once, and we're going to count B once, and in doing so, we've counted this intersecting region twice, so we need to subtract 1 in order to get the union of these 2 A and B events here without double counting. And this is the foundation of probability theory, which seems a bit abstract, but it turns out a lot of useful rules can extend from these axioms. So one of these is the idea of joint probability, so A and B in this case, which is sometimes written like this, probability of A comma B. So if A and B are independent events, then we can compute A and B with the product, or by multiplying individual probabilities here. And so intuitively, so for an example, let's say I want to know the probability of 3 H's, 3 heads. So I could just multiply then the probability of 1 heads 3 times here. So 1/2 times 1/2 times 1/2, which is going to give us 1/8. And if we go back to our original way to figure these problems out of just counting possibilities, so there are 8 possibilities, and one of these is an HHH event. So that also gives us 1/8 there. And then a special case of the third axiom is that if A and B are mutually exclusive, so they have no overlap or intersection here, then we can drop this third subtracted intersection term, and A or B is going to be equal to simply the sum of A and B. So these can be useful. And another useful one is the idea of a conditional probability. And so you'll see this written like this with a bar here. So this is the probability of event B given or based on the premise that A is true. And so this is going to have this definition. So it's defined as the ratio of the probability of A and B to that of just A. So it's this intersection region and the ratio of that to the entire A region here. And so for some more intuition here, let's say I want to know the probability that I get 3 H's, but premised on the fact that we've already done 2 coin flips and gotten 2 H's. So intuitively, this is pretty simple, right? We're basically just asking what is the probability that I get a heads on my next flip, which should be 1/2. But we can also compute it using this expression here, the conditional probability. So the probability of any event involving an H on the end here, so blank blank H, is going to be all of these guys here. That's 4/8. The probability that 2 H's have already occurred here, H blank, is these 2 possibilities here. These are the only 2 that start with 2 H's. So that's 2/8, right? And if we put these together, so 2/8 times 4/8 here, right? We're multiplying these joint probabilities and dividing by 2/8. So this is the probability of A in this case. And that also is going to give us 1/2. And then another useful concept is the idea of conditional independence. So we had this rule of independent results being multiplied together to get their joint probability or conjunction. So A and B is their multiplication. But in practice, many times, A and B, things that you're studying maybe are not truly independent. And so a common workaround is to find some condition which makes them independent. So we might premise their independence on some factor C that's being controlled. So we might say, what's the probability of A and B given factor C here? And then this would then allow us to use this multiplication here. So it's going to be the probability of A given C times the probability of B given C. And that way we've achieved this independence in a case where it would have been not true otherwise. And so that's events, but something we've actually already kind of done without even thinking about it is make use of the idea of a random variable. So a random variable, that's X here. And the idea with a random variable is that we're mapping events to numbers. And so we did this here just kind of intuitively by counting the number of heads. So we had all these 16 events here for the 4 coin toss, but then we said that, okay, this is the 4 heads event, and these 4 guys here are all 3 heads events. And so this number of heads here counts as a random variable. So it's a number that's representing a collection of events here. And so we're no longer having to talk about symbols and letters and events or ideas here, but instead we have a numerical thing that we can work with. And this becomes the basis for frequency distributions and probability distributions that can be visualized using numeric quantities. So we can throw all these things into a histogram here, and we can see that if we plot a random variable on the x-axis, we can then plot the probability on the y-axis and get the shape of our distribution this way. And so this takes us to a really important idea or intuition, in my opinion, which is the idea that the shape of distributions occurs due to degeneracy. So degeneracy is the idea that multiple events can map to the same value, or that in the case of a continuous distribution, that the density of probability is going to be different for different values. So you'll have different events mapping to the same interval, multiple events mapping to the same interval. So the idea here with this discrete distribution is that, we have actually 6 events here that are all mapping to the 2, and that's why we get the peak of this distribution here. Okay, and then, so I mentioned continuous variables, and so discrete random variables are those that you can count, but let's say you have something like, I'm measuring a mass, and a mass is not really countable. It can have any infinite number of values, and those values exist on a continuum. And so if we want to model the probability of such a variable, it no longer makes sense to count events, but we instead think in terms of densities and intervals. So we could have a probability function here, which is based on area under the curve, this density curve here. And if we want to get a value for probability, we have to think in terms of this interval. So from A to B, what is the probability that our random variable takes on a value in this interval? And so you can see here, to get that value, it's essentially taking the area under this curve here. And so that's two basic ideas from probability theory. We have the mass function, probability mass function, or the probability density function, f of x or p of x here. And they're both probability functions where the height of this curve is telling us a probability, and the x is going to be some random variable, some mapping of events to numbers. So some distributions are very well studied and very well known, and one of these is the binomial distribution. And it turns out this is the distribution that we could use to describe analytically and predict analytically our coin flip experiment here. And so a statistical model, a binomial distribution, has an equation. So all of these distributions are going to have a function here. So in this case, this is our p of x, but it's got some more letters here. And so these other letters are parameters, right? So the binomial distribution, if we want to compute a probability, we need to answer some question first. So we need to know how many successes are we interested in. So if it's heads counts as a success here, then our k would be, for example, maybe we want to know what's the probability of 4 k equals 4. We want to know how many trials there are. So in this case, we're doing 4 coin flips, so n equals 4. And then what's the probability of a single success? So in the first example, we said that it was a 50-50 fair coin, but we could imagine a biased coin where you have, say, a 60% chance of getting heads. And then this is going to alter the shape of this curve and alter the results that you get for different values of k. And so this is a very useful distribution. So the coin flip example is a bit abstract, but easy to think about. But in biotechnology, for example, binomial distribution comes up in the case of DNA sequencing. So we could say, let's say we have a DNA sequence, and it's a sequence of nucleotide bases, and we have a machine that's reading those bases, and it has an error rate of 0.01. So there's some chance to substitute a base for the wrong base. And let's say the strand we're reading is 1,000 nucleotides. It's like a long read. And we want to know what's the probability that our strand has k equals 1 error. So in this case, a success is actually an error. And we can use the binomial distribution to model that. And there are other distributions out there. So the Poisson distribution is another example. So this can be used to model things like, let's say I give you a density, a rate of occurrence of events, like buses coming to your stop per hour. And then we're thinking about an interval of time. So in a 10-minute interval, and given that buses occur once every 15 minutes on average, what is the probability that 2 buses comes in my 10-minute interval? So you can use the Poisson distribution to answer this question. And then a biotechnology example. So let's say we have antibodies immobilized on a chip at a density of R equals 10 molecules per square micrometer. So we're using this equation here. And we have a patch of space, T equals 10 square micrometers. And I want to know what's the probability that there are 80 molecules on that patch. So then I would plug and chug into this equation here and then get a probability for that particular question. And then finally, another really common distribution is, of course, the normal distribution or the Gaussian distribution. And unlike the previous two discrete countable distributions, this is an example of a continuous one. And it's going to have parameters which are the mean, mu here, so the center of this distribution, and sigma squared, the variance, or sigma, which is the standard deviation, which is going to indicate the spread of this curve. And if you know these two values, then the shape of a Gaussian distribution is fully defined. And this is, of course, the classic bell curve that is very important for describing experimental error. So this is why we use this so often in science. And you have all sorts of other distributions. So this does not even begin to tap the surface. So you have, for example, the negative binomial distribution, the exponential distribution, the Erlang distribution, geometric distribution. So there are many examples out there of processes that have been studied and have been mathematically proven to be described and modeled by these analytical equations that we have here. And a useful topic in this field is the idea of a cumulative distribution function. And the idea here is that we're summing up as we go. So we're going down the line, going through our x values, and we're taking the probability, not just in a particular bin here, but it's going to be the probability up until that bin that we're looking at. And so the idea is we create this monotonic function where one value on the y here maps to one value on the x, which is not the case here for probability mass or density functions, where a y value here could be, this value of x or this value of x. So these monotonic functions are very useful. In the continuous version, it's the same. So instead of summing to obtain a continuous CDF, cumulative distribution function, you would integrate the density function from -infinity to x. So at each point x, we're taking the summation or the integral of everything that came before it. So at the end of one of these functions, it's always going to be 1 or approaching 1, and the beginning will always start with 0, so from 0 to 1 here. And why is this interesting? So from a scientific computing perspective, one very interesting thing that you can use CDF for is simulation, or the Monte Carlo method, it's called. So we can pick a random number uniformly. So if we have a random number generator, which just generates a number between 0 and 1, perfectly uniform, so you have equal chance to get 0.15 or 0.389. We can choose a random number, and that will map to a particular value x based on the particular CDF we're using. So if we want to sample from the exponential distribution, for example, we could start with the CDF and just rearrange it to solve for x as a function of f here. And so f here is going to be, of course, this y-axis, and we would replace this with a random number. So a random number, say 0.23, we have a parameter lambda, which is the rate or expected value parameter in exponential distribution. So we can plug these things in and get a particular value x, which is going to be randomly sampled from the exponential distribution. So we're transforming the uniform distribution or the random number generator into an exponential distribution generator or sampler. And so here's an example of this in practice. So this histogram here is showing a generated exponential distribution curve, whereas the red here is showing the strictly computed analytical curve. And you can see that they're very close to each other. So in this case, we've done 1,000 samples. We picked a lambda parameter for our exponential distribution. And then I've added this rearranged CDF where we've solved for x or tau in this case. And then we're substituting the probability capital F for u here, which is going to be a random number. And that's going to give us a big list of data that are going to be values randomly sampled from this distribution. So it's as if we've simulated the physical process for an exponential distribution. And so you can imagine an example, like maybe we're modeling the residence time of a protein bound to a surface with an exponential distribution, and we want to simulate the kind of data that might come out of that process. So if we know the rate of unbinding, then we can get these residence times. So that's the Monte Carlo method, and there's another manifestation of the Monte Carlo method, which is the idea of just simulating the process. So let's say we don't know the analytical distribution, but I can clearly describe a process that would generate the correct distribution. So often we don't have access to the exact analytical, mathematical description of the process we're interested in, but we still know how to program something similar. So if we go back to this sequencing example where we have a read and you have some error rate, and I want to know, yeah, what is the probability of particular number of errors for my 1000 nucleotide long sequence? So if we went the route of, for example, writing out all possible events here, like HHHH, HHHT. So this is for 4 coin flips, but now we want to do 1000 coin flips, right? 1000 nucleotides. And the number of possibilities for 4 coin flips is 16. The number of possibilities for 1000 coin flips or 1000 nucleotides is 2 to the 1000, or 1 times 10 to the 301. And so for comparison, for benchmark here, there are 10 to the 80 atoms in the universe. So this is a number that's basically impossible to compute. There's no way we can ever possibly map out all possible events the way we have in this 4 coin toss example. And so a better approach is to sample using Monte Carlo technique. And we could do it using the analytical expression of the binomial function, like we did for the exponential example. But another approach is to just use random numbers to simulate coin tosses. So let's say I have no idea what the formula for the binomial function is, but I can generate a random number between 0 and 1. And if it's a fair coin, we can say, if it's below 0.5, it's heads. If it's above 0.5, it's tails. And I can just repeat this over and over again. So I can make a loop, do this 10,000 times. And you can see that the analytical function here, computed with a binomial distribution, looks almost identical to our simulated example here. And so even though I've only performed maybe 1,000 trials here, instead of the 10 to the 300 possible events, even though I've just sampled this tiny fraction of possibilities, I've nevertheless captured the shape of the distribution very accurately. And so this is a very powerful technique. And so the idea here is we're going to do an exercise. And the premise of this problem is that we're working with a sequence-based experiment, qPCR, and we're using a unique molecular identifier, or a UMI. So this is a random sequence that maybe we would use to identify particular molecules in our experiment. So we're generating random sequences here. And the catch is that our experiment, for whatever reason, is utilizing this restriction enzyme, HAE3, which happens to cut the sequence GGCC. And so maybe this is used for another part of the experiment, but in the context of these UMIs, these random sequences, this is a problem because we're not intentionally putting this GGCC sequence here, but there is a random possibility that it can just appear since we're generating these sequences randomly. And so we might want to know what is the, or we might want to know how many sequences do we need to sample in order to encounter 5 cut events, okay? So we're saying here that 5 cut events, or 5 successes in the language of these distributions, 5 cut events is some sort of critical threshold that we want to avoid. And we want to know how many UMIs are you going to have to sample in order to run into 5 of these events. And so let's simulate it using the Monte Carlo method. And the way you can do that is by generating a UMI, generating a random sequence here. So let's say they're 20 nucleotides long. We can use a snippet of code here to pick a random ATCG nucleotide base letter here and assemble a random sequence. And we want to check then, is GGCC there, and perform this trial many, many times to map out this distribution and keep track of cut events, and in particular, when we hit 5 cut events. And so this is the threshold. And so I haven't told you yet, but there is in fact an analytical distribution function that describes this exact process. It's not the binomial function. And the idea here is that we're doing this without knowledge of that. So we're using simulation to get around the absence of knowing the exact analytical form. So to summarize the lessons of this unit here, we have probability theory. This is the foundation of machine learning. And kind of interesting philosophical intuition is the idea that distribution shapes emerge because of mapping events to the same number. And in general, we have this idea that processes generate events, and those events are how we get probability distributions in the first place. And we can use things like the Monte Carlo method to simulate how those events are generated. And we can also use analytical distribution functions to model those processes. And we can also use those functions to generate or simulate the underlying processes. Okay, and that's it for this unit. So see you in the next one.